These are genetics notes, part 2 for 13, 14, and 15. The next part of notes we are going to talk about are external influences. So we're going to look at nature versus nurture. What affects genes more? Environmental factors can also alter gene expression by ways of cell signaling pathways that ultimately affect gene controls. Now, how does environment actually get in there and affect things? The change in methylation, which is the adding or removing of methyl groups from regions of DNA, may enhance or suppress gene expression. So I've got a couple of pictures over here I wanted to show you so it makes a little bit more sense. Your chromosome, as it's unwound and you've got DNA, has natural abilities for these methyl groups to attach to it. So CH3 is a methyl group. It can attach, see DNA methyltransferase, it can attach to this carbon right here, and when it does that, it attaches to the backbone of DNA, and it can signal for certain genes to be turned on and certain genes to be turned off. What I found is like the more methyl groups you have, the more the genes are turned off, I think typically. So in a normal cell, for example, you might have a methyl group on a couple of places signaling, hey, transcribe this gene, or hey, let's go to this part of the cell cycle, let's stop this part of the cell cycle, hey, cell, you need to die now, your time has come. But in cancer cells, like, it's all turned on and it's all going really quickly, so they don't have the signals to tell them to stop because all of their genes to go are turned on. So the more methyl groups you have, it appears that the more gene turning on you have. So by adding of methyl groups, you can change your phenotypic expression, and the environment can cause methyl groups to be added to your DNA. Genotype plus environment, therefore, leads to phenotype. It's not just your DNA. You do have to have the DNA present. Like, you're not going to express a phenotype for a DNA that you don't have, but at the same time, the environment is going to play a role in how that expression is going to occur. Things that can affect it are seasonal changes, so such as coat color, altitude and plant height, reproduction in water fleas. They can change their sex based on their needs. And psychiatric disorders such as ADHD can all be affected by the environment. So if you have a gene for it, it might not be turned on, it might not be expressing until you go through different environmental factors, such as a bunch of stress can cause methylation of different DNA groups and trigger ADHD attacks and different things. So it's something to think about. How does your environment, and environments can also be foods and chemicals that you're putting on and in your body, how can those affect your DNA? And pedigrees. Pedigrees are basically pictures of genetic phenotypes in families, or inheritance patterns in families. So tracks inheritance patterns in families. Genetic abnormality is considered an uncommon version of a heritable trait that does not result in medical problems. It's just something that's abnormal, such as red hair. It could have been considered an abnormality a long time ago. And then a genetic disorder is a heritable condition that sooner or later will result in mild or severe medical problems. And so you can use pedigrees to track both of these things. And a lot of people are more interested in the genetic disorders because they want to know what the likelihood of them passing it down from one generation to the next. You can use these to determine whether it's a dominant or a recessive trait. So it's used to predict, or used to visualize, inheritance patterns that can be predictably associated with specific phenotypes. So I have a lovely pedigree over here. This is the pedigree of the royal family and their hemophilia disease. So here we have Queen Victoria. She is a daughter of Victoria, and she became a carrier to hemophilia. It's found on the X chromosome. So when her mom's gametes were being formed and making an egg, she inherited a mutated gene. It's only one single base pair that got messed up in the DNA, and it causes the beta hemoglobin. It's caused one amino acid to be replaced with another amino acid, and so it turns the wrong way, and all of a sudden you have a red blood cell that looks like a sickle instead of normal red blood. Well, she had a lot of children, and she only made 2 carriers out of those children, and we know those were 2 carriers, or 2 known carriers, I should say, based on their children. And she also had some that we really don't know their phenotypes. So we're assuming that these guys are normal, or these we know the males are normal. We're assuming this female and this female is normal, but we don't know for sure because they didn't have children of their own. So without knowing their children, we don't know if they would be a carrier or normal. But if you are a carrier, then you have a chance of having a hemophilia positive son. So Mr. Leopold here, he was a hemophiliac. And over here with Beatrice, she also had 2 sons that were hemophiliac sons. And you can see how it tapers on down. And eventually hemophilia kind of was bred out of the royal family. But if you look over, where am I at? There was one cousin group that wed. Oh, here we go. And this male and this female, they are first cousins. And the female was a carrier, and she ended up having 3 boys, 2 of which had hemophilia. So you can kind of see how this traces down. A lot of these we don't know. They were young and all assassinated before they had a chance to breed. And then we had more and more and more and more of them until you get to William and Harry. And based on their phenotypes on down through, you can see that they are no longer carriers of the hemophiliac trait. So let's talk about chromosomal number. Chromosomal number can be seen in karyotypes. They are pictures of the total numbers of chromosomes in an individual. Aneuploid individuals, ANEU, ploid individuals, have too many or too few copies of any one chromosome. In most humans, aneuploids are lethal. They are in less than 1% of individuals. This aneuploids are caused by a non-disjunction, which occurs during meiosis due to the faulty separating chromosomes or chromatids in either anaphase 1 or anaphase 2. So I've got a few pictures of this going on here. We've got, this is a normal male. You can see how you've got a chromosome number 1 for mom, and a chromosome, and it's words failing me. So you've got your sister chromatids where you've got one for mom and its copy. And then you've got your normal X and your normal Y. So this is after replication, but this is not the pair of chromosomes. This is a sister chromatid, so it's original and its copy. This down here is a normal female, and notice these aren't in sister chromatids. These are individual chromosomes before they have been copied or after their copy and after their original split. But you've got number 1 and number 1. So if you look at these, this is more like what it looks like naturally. The bands are where you've got heavier regions of DNA. You've got little points here and here. Those would be the centromeres. And then the bands will line up so you can, they used to just do these and then cut them out and have to match them up literally. So you match up the bands and where they are. So a normal female, they have 2 X chromosomes, and notice there is no Y. In this individual, this is Klinefelter syndrome. This would present as a male, but it would have some secondary characteristics, secondary sex characteristics that are likely female. So it might have wider hips. This individual might not have a lot of facial hair. They might kind of develop a little bit more breast tissue. So overall, slightly more female, but they would have all the plumbing of a male. And then in this individual, they have trisomy 21 or Down syndrome. So they have 3 sets or 3 number 21 chromosomes, and that causes different issues, some physical features, some affecting the ability to learn, like a little bit of mental retardation issues that go along with trisomy 21. Because what happens is you are overproducing genes in this case. So you're producing a gene for this chromosome, and maybe it masks this one, but then you're also going to be producing a gene for the other one as well. And when you have overproduction of genes, you end up with some medical issues. So not only can you have issues with those and chromosome number when you're making meiosis and making gametes, but you can also have issues with chromosomal structure. So the structure of the chromosome itself can have issues. So during prophase 1, faulty crossovers can result in abnormal structure. So again, here's my lovely prophase 1 picture. In a normal crossover, they will split at the same parts, and you will end up with a nice, neat little bit of chromosome 1 for mom ending up on dad's and dad's ending up on mom's. This is how it should look. This was what would make everything happy and healthy. However, in some cases, it doesn't work like that and crossovers are faulty. So I've got a few examples of that over here. A lot of these are called translocations or Robertsonian translocations. So in this one, excuse me, where it splits, you end up with some of this part of it that comes off and it looks a little different. And so this one ends up going on this chromosome. So you have number 12 translocating onto number 7 or 17, and some of number 17 translocating on number 12. That's generally considered bad. You might also have that happening on number 14 and number 21. So you might get some of number 14 crossing over to 21 and 21 crossing over to 14. And then 21 might be split into a few different segments. I actually have my first cousin has a little girl who has Down syndrome or trisomy 21. But it's weird, there's translocations involved. So there's pieces of chromosomes. So she's got like one bottom piece of 120, one of the chromosomes, number 21. She's got a top of another one, and then she's got another piece of another thing. So it's not like it's all cut and dry. So they're not quite sure how her Down syndrome is going to affect her development just yet. Then I've got a few other examples of this translocation stuff. So normal you would end up with one of these and one of these. And then you could have a balanced T, so where one of them comes over to this side, but it balances out. So you lose the bottom, but it ends up on the top of another one. You might have trisomy 21 issues where if you look, see now this one has an extra top here, and then it's got the two bottoms. So now part of chromosome number 21 got taken off and shoved onto this other chromosome, but then you still have the normal number 21s here, so you've got an extra set. Trisomy 13, so you've got two normal 13s, and then you inherit these extras, but this guy right here is the problem, it's extra. Mono 21, so you only inherit one chromosome, and then mono 13 where you inherit two, and then this is the opposite of what would happen, so it would end up with one number 13. This one would end up with 121, and so on. So you see how when it splits, it's got the duplicate up top. After fertilization, you can see its normal thing. So there's a few of them. And again, this is looking at 13 and 21 and how these are Robertsonian translocations and how these can affect an individual. Oh gosh, I'm throwing stuff everywhere. Boom. Trying to make this go away. All right, and I think I had this one more and it was a little bit prettier, but I'm spending some time on this because it's not something that a lot of people know about. This is where 14 and 21 would get a little mixed up. So normal you would have two 14s and two 21s, but you could, so this is your parent, and so this parent, which has a little bit of a dissociation here, if you look at this, it's got this one ended up top. So you could inherit normally, where you have the gametes be a normal one and a normal one. So then you inherit normally. Or this guy could be brought down here, and then you would inherit abnormally. You could get one of these and inherit abnormally. Or you could get just 121. This is lethal. This one's lethal. Trisomy 14s of any sort are lethal. Monosomy 14s are lethal. So these would typically be miscarriages. Trisomy 21, you would inherit an extra set here. Or monosomy 21, if you only get one of those, also is lethal. So not every time when you have extra chromosomes or little chromosomes does it work out. Most of these are miscarriages, and some of these happen so early on in the pregnancy that most women don't even know that they are pregnant yet. I'm going to stop there, and I'm going to start with the next one. later because I've got a mess here.